Hello everyone. In the last lecture of fermentation technology, we have already started with a downstreaming process, and we have studied the few processes included in the downstreaming process. In today's lecture also we are going to study some processes for the product purifications which are carried out in the downstreaming process. Now, product purification is generally done to separate contaminants or the remaining substance or the product from the broth. When the fermentation process is completed or whole the product formation process is completed, it is very important to purify the product in which contains the closely related physical and chemical properties of the products, and different types of contaminants or remaining substances or waste material is also present in the product. So it is very important to purify it, and it is slightly expensive to carry out the product purification process, and it requires highly sensitive and sophisticated equipments for the purification purpose. Now, different types of chromatographic techniques are there, or different equipments also we have to use for this purification process. So this process is slightly expensive, and different types of operations or the processes included in the product purifications are affinity chromatography or size exclusion chromatography, reversed phase chromatography, crystallization, fractional precipitation, or liquid extraction, etc. are the processes used or carried out under the product purification. We will see all these processes one by one. First one is chromatographic technique. Now, this chromatographic technique is used for the separation of our desired product from the mixture, and this mixture is dissolved in different solvents and passing a mixture involved in a mobile phase through the stationary phase. Now, you might have performed the chromatographic technique in which you mix your desired solute or mixture in the solvent, and this it is called as mobile phase, and you run this mobile phase through the stationary phase, which remains in the stationary or in steady state. It may be your paper or the silica gel. It may are used as a stationary phase, and it separates the analyte to be measured from the other molecule in the mixture and allows to be isolated. Generally, for the analysis and the separation of the desired compound in the mixture, the chromatographic technique is carried out. Then the first chromatographic technique is your ion exchange chromatography. Before studying the ion exchange chromatography, there are few terms which we need to understand. First of all, what is ion? Ion is an atom or molecule which has lost or gained one or more valence electron, giving it a positive or negative electrical charges. Now, there may be two types of ions are generally present. One is anion, which is negatively charged, and second one is your cation, which is positively charged. And these both anions and cations are used, and the principle of depending on these electrical charges, the ion exchange chromatography is run. Now, in this ion exchange chromatography, generally a charged stationary phase is used for the separation of the compound, which is also charged. Means here the oppositely charged stationary and mobile phase are used. For example, the resin that carries charged functional group which interact with oppositely charged group of the compound to be retained. Here in diagram you can see. Now यहाँ क्या किया जाता है कि oppositely charged ions are separated. For example, the compound of interest or protein beads which we want to separate or purify, it is positively charged. Then here the beads which are used for the separation purpose should be negatively charged. Means here what will happen when we pass through this our substance or the mixture through this solution, our positively charged product will get bind to the negatively charged beads. For example, if our product is protein, then these our positively charged proteins will get attached to the negatively charged beads. Here you can see the blue color positively charged proteins have got attached to the gray colored negatively charged beads, and remainingly those impurities or the remaining substances which were present in the mixture they have flowed away to the bottom of the column. Means here the negatively charged protein flow through off. फिर यहाँ क्या होता है कि हमारा जो भी पॉजिटिवली चार्ज प्रोटीन था वो नेगेटिव चार्ज बीड्स हमने यूज़ करके क्या क्या सेपरेट किया, और रिमेनिंग जो भी मिक्सचर के कंटेंट थे वो फ्लो हो गए. So here we can easily separate those beads and we will get the pure form of protein by using ion exchange chromatography. Next one is your affinity chromatography. Now name itself indicate there should be some kind of affinity of the mobile and the stationary phase. Now affinity chromatography generally separates the protein of interest on the basis of reversible interaction between it and its antibody coupled to the chromatographic beads. Now here your separating component, your product is protein, and this protein is separated on the basis of its affinity or attachment towards the antibody which is completely or coupled with its interactive antigen. With here the high selectivity, high resolution, high capacity for the protein of interest, purification level in the order of several thousand folds are achievable. Here what is done? The proteins are collected in a purified form or in a concentrated form. By using affinity chromatography, in the pure form we can obtain the proteins. Here the main biological interaction is used for the separation purpose. The antigen and the protein of interest can result to form of various interactions like van der Waals interaction or van der Waals force, electrostatic interaction or hydrogen bonding. And the protein of interest can be removed by simply changing the pH of ionic strength. See in diagram you can see यहाँ क्या किया जाता है, हमारा जो भी प्रोटीन ऑफ इंटरेस्ट है वो प्रोटीन हमारा जो प्रोडक्ट है हमें वो सेपरेट करना है. तो प्रोटीन की हाई एफिनिटी होती है एंटीबॉडी के साथ, तो एंटीबॉडीज उसके रिवर्सिबल एंटीजन के साथ एफिनिटी क्रोमेटोग्राफिक कॉलम में क्या किया जाता है, ऐड किया जाता है. व्हेन वी पास आवर मिक्सचर थ्रू दिस, हियर यू कैन सी द एंटीजन and the antibody specific for those antigen get bind to each other. And our protein of interest also get bind to it due to its coupled or affinity towards the antibody. And after when the remaining material is passed off to and removed through the bottom, After this process, what is done? Just the pH of whole the process is changed, and easily the protein of interest can be separated. Now here the large volume can also be used for the separation of desired protein or our product. But what happens, this process is slightly expensive, so affinity chromatography is generally not used on large scale. And it is highly specific for separation or purification of the protein of interest. Next type of chromatography is size exclusion chromatography. Now name itself indicates these here the separation or the purification of product is carried out depending on the size of the compound. The gel permeation or filtration chromatography, which is GPC, is generally used, and products are separated depending upon its size and low resolution depending on its tertiary or quaternary structure of that particles. See, now here you can see in the diagram there the through the column the particles are different types of beads are set in the column. The porous particle, the small particles can easily move at the faster range and easily separated, and the larger particles remains on the beads or are separated later on. So depending on the size of the particle, the product can be separated. Next one is reversed phase chromatography. The name itself indicates it is the reverse procedure is generally used. In reverse phase chromatography is an elution process used in liquid chromatography in which the mobile phase and is significant more polar than the stationary phase. Now depending on the polarity of the molecule, the separation is carried out. Now in this what happens, the stationary phase is slightly less polar as compared to the mobile phase, and depending on this polarity the separation is carried out. Now what is polarity? The dipole intermolecular force between the slightly positively charged end of one molecule to the negatively charged molecule or negative end of another molecule is known as polarity, and depending on this phenomenon the separation is carried out. Then the next point is your liquid chromatography. Now here the mobile phase is generally liquid and carried out either by this process is either carried out in simple planes or different types of columns, and a special equipment is also used for liquid chromatography, which is your HPLC, means high performance liquid chromatography. In HPLC technique, the sample is forced or forcefully passed through a column that is packed with different types of sporous particles and which acts as the stationary phase, and then the mobile phase at high pressure is passed through it. यहाँ क्या किया जाता है, एक सिंपल कॉलम होता है जिस कॉलम में forcefully porous particles which acts as a stationary phase set किए जाते हैं, and through these particles with high pressure the mobile phase is passed, and depending on its affinity the product is separated. Now here you can see the diagram of HPLC. This is the equipment in which your high performance liquid chromatography is carried out. and all the uh, measures are seen on the detectors and on the data system. So this was all about the product purification by using the chromatographic technique. Thank you.